Good morning. It's April 28th, and this is your daily brief in science. Here's everything you need to know. Archaeologists in Pompeii are using artificial intelligence to reconstruct the face of a victim from the year AD 79 Vesuvius eruption, backed by the Italian Culture Ministry and accompanied by AP imagery. The portrait depicts an older man who died amid debris while fleeing toward the coast, found with a terracotta mortar, an oil lamp, a small iron ring, and 10 bronze coins. The project aims to make archaeological research more accessible and emotionally engaging for the public while staying scientifically rigorous. No name or personal identification is disclosed for the victim, and the article does not detail the AI methods used. The fuller piece notes that methods, datasets, AI models, and the broader historical context are explored beyond this summary. Researchers estimate the individual died early in the 2-day disaster during intense debris fallout, consistent with accounts of people seeking protection with objects. The work fits into a broader trend of AI-assisted research in archaeology and digital humanities. Pompeii remains one of the world's most important archaeological sites and a major tourist destination, attracting millions of visitors. The project seeks to balance public engagement with scientific accuracy, offering a new way to understand one of history's most famous disasters. The study discusses how AI can aid historical reconstruction, museum presentation, and education, while acknowledging ethical considerations and limitations. A new joint effort from RDM Group and Dow unveils CirculaRR, a fiber-based packaging that uses recycled cartonboard with a barrier coating derived from post-consumer recycled plastic to create a fully circular packaging solution. The packaging blends RDM Group's multi-board barrier with Dow's circular polymers and advanced recycling technologies, delivering food contact-capable packaging for items like frozen fish, cakes, fruit trays, and dry pet food. The collaboration aims to bridge fiber and plastic in packaging, accelerating circularity and reducing overall packaging waste. It supports regulatory and tax objectives by increasing recycled content, aligning with frameworks such as the Plastic Packaging Tax and the EU PPWR. The material is drop-in compatible with existing packaging lines, lowering capital expenditure while improving environmental performance. An industry debut is planned at Interpack 2026 in Dusseldorf, with prototypes shown at Dow's booth in Hall 9, C24. The solution is designed to be recyclable within established paper recycling systems and has been tested by 4 Evergreen to verify circularity and recyclability. CirculaRR is ISCC Plus, certified for recycled plastic, and carries ISEGA certification, confirming recycled content and food contact safety. ISEGA certification further confirms suitability for food applications and adherence to safety standards. The product is made entirely from recycled materials, recycled paper coated with advanced recycled polyethylene, and is suitable for diverse food contact applications, including frozen fish, cakes, fruit trays, and dry pet food. Dow highlights advanced recycling as a way to produce virgin-like high-performance plastics from recycled feedstocks, complementing mechanical recycling. The collaboration places CirculaRR within the PPWR framework, noting future requirements for recyclability by 2030 and targets for recycled plastic content to help brands meet obligations. Proteotype is launching a 3-year PhD project at the Medical University of Vienna, funded by Proteotype, with Dr. Yen Tan as the main supervisor and Dr. Klaus Kratochwil as co-supervisor. The aim is to develop circulating proteomic biomarkers for early breast cancer detection and for predicting treatment response, integrating biomarker signatures with clinical data. The PhD student will work within Proteotype's proteomic biomarker platform to measure amino acid-resolved protein signatures in blood, contributing to early detection efforts and monitoring response to CDK4/6 inhibitors. Proteotype's goal is to advance precision oncology by leveraging multi-cancer early detection and personalized medicine tests that improve early detection and treatment decisions through proteomic biomarkers. The HER-Care program is funded by the European Union under Horizon Europe through the Marie Sklodowska-Curie Actions. The consortium includes Karolinska Institutet, University of Cambridge, Institute of Cancer Research, Institute Gustave Roussy, Netherlands Cancer Institute, and the Medical University of Vienna, among others. In addition to training researchers, HerCare aims to push innovation in hereditary and early-onset breast cancer by integrating genomics, proteomics, imaging, and data science. Proteotype Diagnostics Limited has been selected as an industrial beneficiary of the HerCare MSCA doctoral network, signaling leadership in precision oncology research. Joining HerCare represents a strategic step for Proteotype to advance its proteomic biomarker platform toward earlier detection and more precise data-driven treatment decisions. Dr. Emma Yates, Proteotype's co-founder and CSO, welcomes the collaboration, while Dr. Yen Tan emphasizes translating biomarkers into clinically meaningful personalized care. Recruitment for the PhD position is underway, with the selected candidate expected to start in autumn 2026 and the HerCare program funded under Horizon Europe. This work builds on Proteotype's UK intention study, which investigates the blood test's ability to identify patients likely to respond to CDK4/6 inhibitors in the adjuvant setting. A new effort aims to test whether age-related RNA pollution in neurons, combined with genetic mutations, drives neurodegenerative diseases like Alzheimer's, Parkinson's, and ALS, and whether removing that RNA pollution can reverse degeneration. The project seeks to slow age-dependent RNA dysregulation to build neuronal resilience and potentially lead to therapies that protect millions. The overarching idea is that reducing RNA dysregulation in aging neurons could help even when pathogenic genetic variants are present, shifting toward proactive cellular rejuvenation. Co-principal investigators include Douglas Galasco and Jerome Mertens, along with Alex Keim, Fred Gage, and Anne Bang, with teams at UC San Diego, the Salk Institute, and Sanford Burnham Prebys. Collaborators led by Jean Yeo at UC San Diego form a cross-institution team with Galasco, Mertens, Chaim, Gage, and Bang. Promising candidates will be tested in 3D brain tissue models called ispheroids, and then in animal models to evaluate potential effectiveness for humans. Leading therapeutics will be evaluated in ispheroid models that mimic brain tissue architecture, linking cell culture to in vivo studies for efficacy and safety before clinical progression. The project reflects state support for high-risk frontier science in regenerative medicine, aiming to alter chronic brain disease trajectories through discovery and clinical research. Robotics-enabled high-throughput screenings will identify thousands of potential treatments, including FDA-approved small molecules and targeted RNA therapies, capable of reducing RNA pollution and restoring neuronal health. The screenings will evaluate compounds for their ability to clear RNA pollution and rejuvenate neuronal function, prioritizing candidates with translational potential due to prior safety data. The collaboration among UC San Diego, the Salk Institute, and Sanford Burnham Prebys has secured a 4-year $13 million CYRM grant to explore reversing age-related neurodegeneration by eliminating RNA pollution in aging neurons. The project aligns with CIRM's DISC-4 portfolio, part of an $80 million commitment to 6 California researchers pursuing foundational science with translational potential in regenerative medicine. A major breakthrough in neurodegenerative disease genetics has realigned 2 long-separated research tracks. Rosa Rademakers, a dementia researcher at VIB and the University of Antwerp, shared the 2026 Breakthrough Prize in Life Sciences for co-discovering that an abnormal expansion in the C90RF72 gene is a primary genetic cause linking ALS and FTD. That finding, first clarified through Mayo Clinic work in 2011, shows affected individuals can have hundreds or thousands of repeats in C90RF72, far exceeding levels in healthy people. The discovery unified ALS and FTD under a common genetic mechanism, reshaping how scientists approach disease pathways, biomarkers, and therapy development. Trainer, co-recipient, and senior investigator at the National Institute on Aging highlighted patient involvement and the use of DNA samples and clinical data to translate research into patient impact. The prize underscores how this shared mechanism has accelerated genetic testing, counseling, and the progression of several therapies into clinical trials. The C90RF72 mutation now accounts for about 1/3 of inherited ALS and FTD cases in Europe, enabling families to pursue genetic testing and informing research directions. RAID makers and trainer independently identified the same genetic abnormality in 2011, tying the two conditions together through a rare DNA sequence expansion. The work has since influenced a broader research landscape, with clinicians and researchers collaborating on disease mechanisms and inheritance patterns. For those seeking deeper context on FTD research, resources like AFTD's FTD Science Digest profile scientists and their work advancing understanding and treatment. This has been your daily brief in science. To read more about these stories, follow the links in the episode bio. You can also subscribe to these updates via email at www.brief.news. For more daily podcasts about the topics you love, visit brief.news/podcasts. Tune in tomorrow. We'll be back with everything you need to know.